Bismillah ar-Rahman ar-Rahim. About the muscle tissue, one of four basic tissue in our body. In the muscle tissue, the cells within this tissue specialize for contraction, and the muscle tissue in our body is classified according to the morphology and function into three groups of muscles: skeletal muscles, cardiac muscle, or smooth muscle. Another name for the skeletal muscle is striated and voluntary muscle, and for the smooth muscle, the another name is a visceral muscle. Here we have two prefix for the muscle. Myo means the muscle, myofiber means the muscle fiber, and myofibril means the fibril of muscles. Another prefix is sarco. Sarco usually used for connective tissue and also for the muscle. Here for the muscle, for example, sarcolemma, sarcoplasm, and sarcoplasmic reticulum. Sarcolemma stands for the cell membrane of muscle cell, sarcoplasm the cytoplasm of muscle cell, and sarcoplasmic reticulum stands for smooth reticulum endoplasmic within the muscles. Under the microscope, this muscle cell, including this picture you can see here, for example, for the skeletal muscle, under the microscope we can see very long cell, so we call them muscle fiber because they are very long. The cells have a cross striation. You can see this cross striation here. They are multinucleated, and the nuclei are located in the peripheral position. This muscle contract very quickly and very forceful, and they are contract under our voluntary control. Another picture is related to the cardiac muscle cell. They are enlarged cell. They are branched. You can see, for example, at this end cell branch to three branch, and in this end you can see the cell has two branch with central one or sometimes two nuclei. The cell have a junction with each other at the site called intercalated disc. Again, here we have a cross striation under the microscope. The cardiac muscle cell contract involuntary, vigorous, and rhythmic. Smooth muscle cells are fusiform, lack striation, one nucleus in the central position. Contraction in these cells also are involuntary and very slow. Let's start with skeletal muscle. This cell created during the embryonic development by fusion of some cells called myoblasts. After fusion, create the myotube. Here explain why these cells have multinuclear, because of fusion of several myoblasts. After formation of myotube, the myotube differentiated by producing a lot of actin and myosin. The cells filled the cytoplasm with this myofilament and displaced the nucleus against the plasma lemma. And after differentiation, the myotube create muscle fiber. You can see some of cells, some of myoblasts don't fuse to the myotubes and remain adjust to the myotube and formed satellite cells. Actually, the satellite cells here function as a stem cell and participate in the regeneration, the muscle injury repairing, and sometimes, especially in the athletic person, to growth of muscle in two-way hypertrophic and hypertrophic and hyperplasia. Both of them, in the hyperplasia, satellite cells proliferate and create a new muscle fiber. In the hypertrophic, the satellite cell joined to the pre-existing muscle fiber and create the new muscle fibers with large diameters. Like the nerves in the nervous systems, here connective tissue organize muscle cells. Like the nerve, the connective tissue surround each muscle fiber, here called endomysium, like endoneurium. Muscle fiber within the muscle creates some muscle fascicle, and the connective tissue surround each muscle fascicle and called premisium, and the entire of the muscle surrounded by a connective tissue called epimysium. So around the whole of muscle, epimysium. Around the fascicle we have premisium, and around each cells we have a endomysium. This connective tissue supply the nerve and blood vessel for the muscles, and also this connective tissue have a very important function. I will explain in the next slide. For better understanding, for example, imagine we have a object with a weight of 2 tons, and we want to pull it on the surface of this table. We have a 6 person to pull it. For using the power of all of these person, usually we use a rope. Connect the rope to the object, and all of person grab the rope and pull it on the surface of table. Actually, in the muscle, all of the cell attached together like a rope, like a function of rope here, by a connective tissue, and all of this connective tissue, endomysium, premisium, and epimysium, emerge from the end of the muscle as a tendon and attached to the bones. So by this way, we can use the power of or the force created by all muscle cells, and we can pull the bone by this way. Again, here you can see around the whole of muscle we have a epimysium. Around the fascicle we have a pre, and around each cell we have a endomysium. Let's see in the real picture. For example, here you can see we have a cross section from the whole of the muscle. So the connective tissue around the whole of muscle called epimysium. You can see the fascicle. The white area around each fascicle is the premisium, and around each cell, the light area around each cell called endomysium with more magnification. For example, here you can see multi-fascicle. So around each fascicle, this part we have a premisium, each cell endomysium, and all of the muscle we have a epimysium. Here, with preserving the connective tissue, because all of this picture will lose the connective tissue by fixation and tissue processing, but here we preserve them. You can see a epimysium and premysium, and around each cell we have a endomysium. Muscle contain a lot of blood capillaries supply the nutrient and oxygen for the muscles, because the function of the muscle need more nutrient and the oxygen. So these blood vessels, a high density of capillary here. For example, you can see the capillary in this picture supply these materials for the muscles. And below picture indicate the junctions border between the tendon and the muscle. When we magnify it, you can see the muscle fibers ended in this part, and from this part started the tendons. The junction between the tendon and the muscle should be very tough and should be very firm. These two tissues should be firmly attached together. When we magnify, with more magnification of this part, we can see a folding of the cell membrane at the end of the muscle cell to increasing the contact surface between the tendon and the muscles. With more magnification, under the electron microscope, we can see with high resolution this folding. If I highlight the same cell membrane in yellow, we can better see the cell membrane folding in this part. The collagen, especially collagen type 1 from the tendons, insert within these foldings and firmly attached to the external lamina of muscle cells, and prevent detachment, easily detachment of tendon and muscle cell. This part called myotendinous junction. In the cross section from the skeletal muscle cell, we can see polyhedral or polygonal cells. Also indicate they are multinucleated. For example, here you can see these cells have 3 nuclei in the peripheral position. We cannot see cross striation or striation in the cross section from the skeletal muscle cell. We can see an endomysium around each cell. Around each fascicle we have a premysium. Again, here cross section indicate the endomysium, multinucleated, peripheral position of the nucleus with more magnification, peripheral position, multinucleated, and endomysium. The nuclei within the endomysium, they are a scattered fibroblast within the endomysium. But in the longitudinal section from the muscle, skeletal muscle cell, we can see cross striation. Again, the number of the nuclei in the peripheral position. Again, endomysium. Here we can see better. Premysium. The next slide also we can see again the striation, multinucleated, endomysium, and also we can see some cells within the endomysium as the fibroblast. The striation under the microscope, we can see alternative dark and light line. The dark line called A band because they change the direction of polarized light, comes from the anisotropic, and for the light part we called I band. comes from the isotropic. Don't change the polarized light. With more magnification, for example, here you can see the A band and I band. When we magnify more, we can see the I band bisected by another dark line called the Z disk. Z comes from the zwischen. It's a German word, means the between. So each I band divided in two equal parts by a Z disk. Under the electron microscope, again we can see Z disk, I band, A band. With more magnification you can see again the A band also have a light area in the middle part. This light area, this bright area called H zone, comes from the hela, means again the German word means the bright, lighter part. And in the middle of the H zone we have another dark line called M line. From the word Mitte means in the middle of A band. From one Z disk to another Z disk called sarcomere. Mere means unit, sarcom is muscle, means subunit of muscles. By repetition of sarcomeres end to end in the muscle cell create myofibrils. I will show you better in the next slides. Why the muscle under the microscope have this kind of features? For example, dark part, light part, dark line, or something like this. All of this is due mainly to the myofilament, syn and sig filament. Regular organization or arrangement of this myofilament within the muscle fibers create some feature like these: I band, A band, Z disc, H zone. All of these property created by the arrangement of this filament. For example, here you can see in the Z disc we have some protein attached to sarcomere together. In the I band we have just a syn filament. In the A band we have a sig filament in addition to overlapping with some part with the syn filament. And in the H zone we have just sig filament. Again, in the M line we have some protein hold the sig filament in place. Now we want to explain in more detail the structure of sig and syn filament. It starts with the syn filament. The syn filament created by a G actin. G actin means globular actins. By polymerization of G actins, they create F actin, fibrillar actin. Two strands of fibrillar actins twist together and make a syn filament, like this. Each G actins, you can see, have a dark spot on each G actin. Is actually myosin binding site. It means each G actin can bind to the myosin. We have some associated actin protein. Actually, they are a regulatory protein. For example, here we have two of them, tropomyosin and complex of troponin. You can see tropomyosin is a two polypeptide twist together and located at the groove between the two strand of actin. And complex of troponin, when we magnify, we can see this complex has three components: troponin T, troponin C, and troponin I. We call them troponin T because this component attached to the tropomyosin. You can see the red part. Troponin C because this component attached to the calcium. And troponin I regulate the interaction between the myosin and actin. The Sikh filament also is formed by polymerizations or by assembling of myosin molecules. Myosin molecules has two heavy chains and two pairs of light chains. Heavy chains actually they are have a rod-like shape with a head and tail. The tail of two heavy chains twist together, and each head has a two part, one part for a actin binding called actin binding site, and another part for a ATP binding. And also this part have a ATPase activity, can hydrolyze the ATP. By assembling of several hundreds of myosin molecules, we have a Sikh filament. In the Sikh filament, you can see we have a overlapping of tail part, and all of the head goes out of the Sikh filament, and usually toward the ends of Sikh filament. We can see in this part the tails assembled and overlapped, make a road part of Sikh filament, and heads you can see go out. They are ready for attaching to the actins. Again, here you can see all of the heads toward the end of Sikh filaments from this port and this one. With more magnification, you can see a band here. In the A band, we have overlapping part between the SYN and Sikh filament, and the middle we have a H-zone. Actually in the H-zone just we have a Sikh filament without overlapping, and the M line we said we have some protein concentration here. By end-to-end repetition of the sarcomeres, we have a myofibrils. For example, sarcomer attached to another sarcomer, and continue of these junctions make a myofibrils. Myofibrils filled the cytoplasm of the muscle fibers, and you can see displaced the nucleus against the cell membrane. When we have a section, for example, from the A band overlapping part, we can see each Sikh filament surrounded by a Sikh SYN filament, actin. When we have a section from the H-zone, you can see we have just Sikh filament. In the M line, you can see we have a protein connect the Sikh filament together. And also in the I band, you can see each SYN filament surrounded by 6 another protein called titinal will explain. The muscle cell called muscle fiber, and this Sikh and SYN filament called myofilament. In addition to previous protein I explained, Here we have other proteins are very important in the structure of the myofibrils. One of these protein is nebolin. You can see a nebolin located along the symphylamen, the actin. This protein attach the symphylamen to the Z disc and also determine the length of symphylamen. Without nebolin, the polymerization of the actin filaments continues and disorganize the myofibril or the sarcomer structures. But nebolin prevent this polymerization and determine the appropriate length of symphylamen. Another protein here you can see attached the SIG filament to the Z disc called titin. It's the biggest protein in our body and provides elasticity for the sarcomers and also they stabilize the myosin by attaching them to the Z disc. We have some protein in the M line, for example, called myomysin and C protein. These two proteins hold the SIG filament in the place in the middle of the sarcomers. In addition to these two proteins, we have another protein in the M line called creatine kinase. The creatine kinase transfers a phosphate group from the phosphocreatine to the ADP and supply the ATP for muscle contraction. This protein or this enzyme located in the M line. Aggregation of these protein in the M line create a dark line under the electron microscope. About the muscle contraction and the mechanism of this contraction. In the muscle contraction, neither SIG filament nor symphylamen change their length. We don't have shortening of this filament during the muscle contraction, but we have a sliding of these filament over each other, so-called the sliding filament theory. Actually, the symphylamen slide over the SIG filament. For example, here you can see Z disc in the green, SIG filament, myosin and symphylamen acting. During the contraction, you can see the sliding of symphylamen over the SIG filament. This sliding started by an ion called the calcium ions. When we have a calcium ions within the cytoplasms, the calcium ions attached to the troponin complex, troponin C, TNC, and attaching the calcium cause some conformational changing in the structure of the troponin, and this changing in the shape of troponin you can see because it's attached to the tropomyosin, and this changing of a structure you can see change the position of tropomyosin and expose the myosin binding site on the G actins. So the head of the myosin can attach to the exposed myosin binding site of G actins. After attaching, now we have some conformational change in the head of the myosin. This called pivot. This pivot pulling the actin filament toward the M line and cause shortening of sarcomers. After attaching the new ATP to the head of myosin, we have a detachment. So the muscle contractions created by several repetition of attachment, pivot, and detachment cycle. For example, here you can see the myosin sighting bind covered by a tropomyosin. After exposing this area, you can see myosin head attached to the G actins. Then we have a pivot, changing the structure of head of myosin, and this conformational changing, pulling the actin myosin, or cause the sliding of actin myosin over the actin filament over the myosin filament. You can see this sixth here: attachment, pivot, and detachment. By exposing, we have attaching the head of myosin. Then we have a pivot, or changing a conformational change of head of myosin, and pulling the actin filament. By attaching new ATP called detachment of head of myosin. And again, repetition of these cells. After this, because the mitochondrial activity is stopped, we don't have new ATP to detach the head of myosin, so create rigor mortis, the rigidity of muscle after this. In the dead body, usually after this, we have this rigidity called rigor mortis, because we have an attachment and we have a contraction in all of muscles. Here you can see the muscle in the relaxed position. We have an I band and A band. When we lifting a very light weight, we have a partial contraction. The length of I band decreased. The length of H zone decreased. The overlapping area increased. And in the full contraction, we don't have any I band, we don't have any H zone, because overlapping area complete by full contraction, and you can see the Z disc attached to the A band. So we don't have any I band in the full contraction.